Meiosis follows the same general process as mitosis, but let's talk about some of the key differences. To begin with, in both mitosis and meiosis, we start with replication of chromosomes. And so we start with sister chromatids. In mitosis, sister chromatids are separated, and that's it. This means that each cell gets an identical copy. Each cell has one of each of the homologous chromosomes. In meiosis, replication produces those sister chromatids, and then we have two cell divisions. In the first cell division, homologous chromosomes are actually separated. So this is something that does not happen in mitosis. In the second phase of meiosis, and that's our other major difference, a second cell division. In the second phase of meiosis, sister chromatids separate. And finally, we get the last major difference, and that is that meiosis produces 4 daughter cells, whereas mitosis only produces 2. So let's look at meiosis with respect to those chromosomes. So we start with a pair of homologous chromosomes. So this one, in this cell, one of those homologous chromosomes would come from mom, the other from dad. Keep in mind that if we're talking about eukaryotes, there's probably multiple chromosomes. So if we're talking about humans, we have 46 chromosomes, 23 pairs of homologs. Each of those homologs is duplicated, so we now have our sister chromatids, but the same number of chromosomes. Meiosis I separates those homologous chromosomes into 2 daughter cells. Each daughter cell has 1 copy of each of the homologs. Then in meiosis, actually, before we move on, at that point, we have reduced the number of chromosomes in the cell. So after meiosis I, we go from diploid organisms to haploid organisms, organisms that only have 1 copy of each type of cell. So meiosis I reduces the number of chromosomes in a cell from 2n to 1n. Then Meiosis II separates sister chromatids, and we'll see that Meiosis II is essentially identical to the process of mitosis, where sister chromatids separate. No more reduction in chromosome number, though, although it looks like we have reduced the number. We've only separated our sister chromatids. Remember, sister chromatids are counted as 1 chromosome. So now at the end of meiosis, we have our haploid daughter cells. So let's look a little bit more detail at the process of meiosis. The stages and the sort of mechanics of meiosis are almost identical to mitosis. And so we're not going to go through each of the phases again. Prophase, just really quickly, chromosomes condense, microtubules form. Metaphase, chromosomes line up at the metaphase plate. Anaphase, they're pulled apart. Telophase, the chromosomes decondense, the nucleus reforms, and the cell performs cytokinesis. This happens twice, though, in meiosis. So once meiosis I produces 2 daughter cells, then each one of those daughter cells divides, and that's where we get our total number of 4. Keep in mind that meiosis, the purpose of meiosis is reduction in the number of chromosomes. So let's talk about some of the differences, some of the specific differences at the DNA level that happen between meiosis phase 1 and meiosis phase 2. Again, I'm not going to talk about every single step in this process. I'm going to focus on the one that the differences between mitosis and meiosis. So in prophase 1, we see our first difference. In prophase 1, our homologous pairs of sister chromatids actually pair up. they associate together, which never happens in mitosis. In mitosis, homologous chromosomes have nothing to do with each other. They're separated independently of all other homologous chromosomes. But in Meiosis I, in order to correctly partition those chromosomes and to eventually reduce the number of cells, homologous chromosomes actually join up. And they join up through a process called crossing over. Crossing over produces a physical link between sister chromatids. And in this process, those sister chromatids actually exchange some genetic information. We'll talk about that process on the next slide. Another difference is in metaphase 1. So in mitosis and in mitosis and meiosis, all the chromosomes line up at the metaphase plate. The difference here is because of crossing over and those sister chromatid pairs bound together, they line up as a set of two homologous chromosomes, as sort of a unit. And again, we don't see that in mitosis. If you look at this picture, though, you can see that they are organized in a way that this cell is going to get 2 red and 1 blue. This cell is going to get 2 blue and 1 red. So they can line up in different combinations of chromosomes that came from mom and chromosomes that came from dad. Both crossing over and this assortment of chromosomes is going to be important later, and we're going to talk about those. So let's talk about the process of crossing over real quick. In prophase 1, homologous sister chromatids, so that is 2 pairs of sister chromatids, homologous chromosomes with their sister chromatid, line up together and physically exchange pieces of DNA. The purpose of this is to hold those chromosomes together so that they can move to the metaphase plate and be partitioned correctly. That's why it happens. The consequence of this, though, is that it produces genetic variation. So if you look at this picture here, remember I said we have these proteins called cohesins that hold those sister chromatids together. We talked about that in the mitosis video. Hold them together. You can almost see the zipper-like protein there. And then the 2 of the DNA strands between the sister chromatids are actually broken and sort of flip-flopped with the other chromosome. So we can see that over here. We see that this blue chromosome now has a piece of red chromosome on it, and this red chromosome now has a piece of blue. This is called crossing over. And in crossing over, we form these X-like shapes between our 2 sister chromatids. So I'm actually going to go back to the previous slide. We can see that here as well. We see crossing over right here. That point of crossing over is called the chiasmata. Just a fancy way to say the X shape that the chromosomes make. And when that happens, you can see that this chromosome here actually has picked up, this red one has picked up a piece of blue. And you can't see it, but that blue chromosome has picked up a piece of red. You can see it a little bit over here on this chromosome. It's actually that blue chromosome now has some red. So what's happening here is that chromosome from dad is exchanging information from chromosome from mom. And so you get unique combinations of chromosomes that originally came from 2 different parents. And that's a really important consequence of crossing over. So the purpose is to correctly move and separate chromatids and homologs, but the consequence is genetic variation, and we need genetic variation for evolution. Continuing on with Meiosis I, the anaphase and telophase phases are essentially the same, except that anaphase 1 pulls apart homologous chromosomes, and sister chromatids remain attached Again, that's our difference from mitosis, as we separate homologous chromosomes. And then at telophase and cytokinesis, that's where we actually divide our cells, and now our cells are haploid. We've gone from 6 total chromosomes in anaphase to 3 in each cell. So we have cut our number of chromosomes in half. Next, we get to Meiosis II. So the process occurs all over again. Telophase at the end of Meiosis I completely finishes, the nuclei reform, the chromosomes decondense, and then Meiosis II, everything starts over again. Another difference here is that Meiosis II does not have a DNA replication step. So it doesn't re-enter the cell cycle and replicate its DNA. It just stays within the meiotic phase. At the end of anaphase, due to crossing over, we are going to produce 4 cells that are genetically different from each other. So no 2 daughter cells are the same, and that is due to that process of crossing over. And you can really see in this picture, this anaphase picture, where that exchange of genetic information has occurred. So now we have these new novel chromosomes. And then again at the end we have 4 daughter cells, and this is different from mitosis that produces 2 daughter cells. So the goal of meiosis is to produce gametes and to produce genetically different gametes from one another. There's a really nice summary sheet on page 209 in your book. I'm not going to go through it, but I would say that there is a 100% chance that in the future you will need to describe the differences between mitosis and meiosis. So I would strongly suggest that you take note of this page. So sexual reproduction is really important in that it produces genetic variation in a population. And there are several sources of genetic variation in the process of sexual reproduction. So I've highlighted one of them here. The first that we're going to talk about is independent assortment. And keep this phrase in your mind because we're going to come back to it next week in chapter 11. In independent assortment, chromosomes randomly orient themselves toward one pole or the other. So this can happen at any combination of red-blue. So here we have red, red, blue, and then the bottom cell is blue, blue, red. We could also have red, blue, red, and blue, red, blue. Each of those combinations is producing new varieties of new combinations of whole chromosomes. So some chromosomes that originally came from dad and originally came from mom are being mixed up and recombined when the ovaries of their children are producing eggs. Same thing with the sperm. So their grandchildren will be a combo of each of the parents' genomes. So this gives you new combinations of entire chromosomes. So that's one way that each of those cells is genetically different. Next, we have that process of crossing over. So in this process, not only have we shuffled the combination of chromosomes from mom and dad, but we have also shuffled the individual chromosomes. So we've exchanged genetic information between mom and dad. So mom and dad produce an egg, and then that egg grows into a boy, and now that boy is producing sperm. We've got new combinations of his parents' chromosomes and combinations of parts of the chromosomes. So we get very different gametes in children compared to their parents. The last part of, or the last source of genetic variation in sexual reproduction is random fertilization. So if you think about the millions of sperm that are produced, each are genetically different from one another, and then any one of those sperm can fertilize basically if you're thinking about it statistically, any egg in a population. So if you have two unrelated individuals combining their genetic material, now you have even more genetic variation. You have variation from independent assortment, variation from crossing over, and now variation from that random fertilization. So there's all kinds of new genetic variation that can occur in sexual reproduction, and this allows a population to be able to adapt and change if there's a new environmental pressure. It allows for evolution to work on a population. So the reason that we care about all of this genetic variation is evolution. Sexual reproduction is important because it increases variation. Genes are mixed and matched, and sometimes mutations are even introduced, and those mutations are passed on. A population can adapt to environmental conditions because some individuals are better suited for their environment because of their combination of genes than others. Kind of the definition of evolution. Some individuals are better suited for their environment because of their heritable traits. Evolution can only work if there's existing variation in a population. So if a population has no variation or doesn't have variation that allows it to survive, then it's not going to survive for the next generation, or it's not going to be very successful. So keep all of these things in mind for when we get to our evolution unit, because we're going to come back to this quite often.